I'd like to ask Ravi Salgia to come up and describe the winner, please.
Thank you, Steve and Anthony, and thanks to the chairs as well as the board for letting me present, but I won't reveal the name. And it's a humble beginning for this individual, and this individual started in medical school in actually University of Kashmir at the Government Medical College of Srinagar, but was a house officer at the University of Kashmir as well, and then transferred over to the United States. And this individual did his residency at the Sisters Hospital at SUNY Buffalo from 91 to 94, and was a Hem/Onc Fellow at the NCI as well as the NIH from 1994 to 97. Then went on to do a BMT, a bone marrow transplantation fellowship over at Georgetown, and was an instructor at Georgetown, but then ultimately moved to Oklahoma as an assistant professor. I think that's when this individual started to make a career out of doing research as a clinician scientist, especially research in the context of mesothelioma. And this individual has published incredibly and immensely, as well as some brilliant papers in the context of mesothelioma. Once this individual moved to as an investigator at the Laboratory of Molecular Biology and the Center for Cancer Research at the National Cancer Institution from 02 to 09, and then became the chief of solid tumor in immunotherapy section from 06 to 13, but then ultimately became tenured as a senior investigator in the LMB at the NCI as well as the Center for Cancer Research. Most recently, in 2013, this individual actually became the chief of thoracic and solid tumor immunotherapy, which is quite an honor at such a young age, but then has done brilliantly in the context of pleural mesothelioma, peritoneal mesothelioma, has recently published a paper that's very widely quoted with it will be a high impact factor as well as an H-index in Science Translational Medicine in 2013 for mesothelion immunotoxin, and has published in PNAS, Cancer Research, Clinical Cancer Research, and also most recently had been awarded the Pioneer Award from the Mesothelioma Foundation, has served on many boards, many scientific advisory boards, and has reviewed an incredible number of grants and is very prestigious in terms of awarding these grants for other individuals as well as mentoring other individuals. And I'm going to stop it here, but really I'm impressed by the career of this individual, and it's a very nice honor for me to present that.
So some of you may have already worked out who this person is, but for those who haven't, I have great pleasure to announce the 2014 winner of the Wagner Medal is Raffit Hassan. One thing before he talks is that he's not going to get away with just saying thank you, but he's actually going to give a talk as well.
Thank you so much. It's indeed an honor to get this award, especially when one considers the past recipients of this award, and would like to thank iMig for recognition of our work at NCI. And it's especially nice to get this award in South Africa, where Professor Wagner made the seminal discoveries of mesothelioma and asbestos exposure. So I'll be talking about our work over the last many years to develop mesothelion-targeted therapies for mesothelioma and cancer, and provide a history of our work and what the future looks like. In case you have trouble figuring me out, I'm here at my lab bench in the Laboratory of Molecular Biology at the NCI, which was established by Ira Paston. So this is a very famous lab at NCI. A couple of his postdocs went on to win the Nobel Prize, including our present boss of NCI, Dr. Wormus. So I went to see Ira and interviewed with him somewhere in 1995, fall of 95. I was looking for a lab to work in. So I met with Ira in his office, and he gave me a paper, said that we have this paper, we're just submitting for publication, and that mesothelin could potentially be a good target for cancer therapy. And if I joined his lab, maybe this is something I could work on. But he didn't tell me that I would be working on it for the rest of my life. So mesothelin is a cell surface glycoprotein. Its expression in normal human tissues is limited to the mesothelial cells that line the pleura, peritoneum, and pericardium. It's made as a precursor protein, which is cleaved by the protease furin to a 31 kilodalton shared fragment, which was independently identified by Yamaguchi and his colleagues, and they call it MPF because it had megakaryocyte potentiating activity in mouse bone marrow cultures, and the 40 kilodalton fragment, which is attached to the cell membrane that we call mesothelin. We and others have shown that mesothelin is highly expressed in many cancers, including almost all epithelial mesotheliomas, majority of pancreatic cancer, about 70% of ovarian cancers, 50% of lung cancers, and other cancers. So the approaches that we are using, we believe, will be useful for treating many common solid tumors. As you heard, Ingegerd Helmström, Bruce Robinson, and Jeanette Craney did pioneering work and showed that mesothelin levels are elevated in patients with mesothelioma. Ingegerd Helmström had previously shown that these levels are elevated in patients with ovarian cancer. But surprisingly, we do not see elevated levels of mesothelin in patients with pancreatic cancer, despite the fact that most of these tumors are mesothelin positive, and we're not sure why that is. The biologic function of mesothelin is not clear at this time. Mutant mice that lack the mesothelin gene showed no phenotype and reproduced normally. We and others have shown that mesothelin is a novel CA125 binding protein and may play a role in tumor implantation in the pleura and peritoneal cavity. Recently, we published that MPF and mesothelin stimulate the growth of the lung cancer cell line A549 in the peritoneal cavity of mice. This is the first in vivo demonstration of the function of mesothelin and MPF. However, the majority of our work has focused on targeting mesothelin for cancer therapy. The high cell surface expression in many solid tumors makes it a good target for antibody-based therapies. And expression on the pleura, pericardium, and peritoneum was a concern when we thought about using mesothelin for targeted therapy. This is one of my first papers I published looking at an antibody to mesothelin called K1. And in this study, we implanted mice bearing mesothelin negative tumors and mesothelin positive tumors and showed that the radiolabeled antibody localizes to these tumors in mice. It took us another 15 years to show in patients that in fact this is true. This is a study that we just did with an antibody you've heard about, emotexumab, also called MORAb-009. It's a humanized antibody to mesothelin. So we labeled it with indium-111. So this is one of my patients with metastatic mesothelioma to the inguinal nodes, as you can see on the PET scan. We did a biopsy, it highly expressed mesothelin. And once we injected radio-labeled indium-111, it localized the tumor. So this was an important proof of principle study to show that these antibodies do in fact target the tumors. So there are different approaches to target mesothelin, but I'll be focusing the majority of my talk about targeting mesothelin-expressing cancers using an immunotoxin, and towards the end I will briefly talk about other approaches to target mesothelin. As you know, very few antibodies by themselves are capable of killing cancer cells. There are some notable examples such as trastuzumab or Herceptin or rituximab. When they bind to the tumor cell, they interfere with some important signaling pathways and the tumor cell dies. But the majority of the antibodies to tumor antigen do nothing. The antibody just binds to it, but the tumor cell does not die. To make these antibodies useful for cancer therapy, there are various things we can do to allow them to target the protein of interest but have a payload attached to them. One approach is to attach protein toxins to them. These are called immunotoxin. The other approach is that you attach highly potent chemotherapy drugs that you cannot otherwise give intravenously, and these are the antibody drug conjugates, and some of them, such as the Herceptin, DM1, has been approved for breast cancer. And the third approach is that you attach radionucleotides to the antibodies, and these are useful for hematologic malignancies. So our work is mostly focused on this approach. So a recombinant immunotoxin consists of an antibody or an antibody fragment. This is the FV, which recognizes the antigen, linked to a toxin. So the toxin that we use is called Pseudomonas Exotoxin A, which is made by the bacteria Pseudomonas aeruginosa. The toxin has 3 binding domains, and these are important as I will come back to it towards the end of my talk. The toxin has 3 domains: a binding domain that allows it to bind to the cell membrane, processing domain, or domain 2, whose function we don't know, and shown in the red is the main catalytic domain, which ADP ribosylates elongation factor 2, leading to inhibition of protein synthesis. So to make an immunotoxin, we get rid of this binding domain, replace it with the FV, and you make an immunotoxin as a single protein. Once the immunotoxin binds to the antigen, in this case mesothelin, it is taken up by clathrin-coated pits, undergoes processing in the endosomes, ultimately goes to the cytoplasm, inhibits elongation factor 2, leading to inhibition of protein synthesis and cell death. So SS1P is an anti-mesothelin immunotoxin, which has high affinity for mesothelin. It is cytotoxic to mesothelin-expressing tumor cells from patients with mesothelioma and ovarian cancer, and causes regression in nude mice. So this is one of the initial studies I did when I was at University of Oklahoma, where we did what we call as organotypic or 3D cultures. So these were samples from a patient with ovarian cancer. So we took the tumor sample, grew it in collagen gels, and treated with different doses of the immunotoxin. As you can see, there is dose-dependent killing of the tumor cell, as well as increase in apoptosis. Similarly, looking at mesothelioma cell lines from patients with peritoneal mesothelioma, we showed that they highly express mesothelin and are very sensitive to the immunotoxin. Based on these preclinical studies, we initiated a phase 1 clinical trial of SS1P. This was for patients with mesothelin-expressing cancers who had failed standard therapy. We established the maximum tolerated dose, and the dose-limiting toxicity was pleuritis, which was something which we expected because the normal pleura expresses mesothelin. However, there was no activity. There were no responses seen in this trial. And the main reason was that SS1P was immunogenic. So it's a foreign protein. It's a bacterial protein. So the patients developed antibodies, and about 90% of the patients developed antibodies after just 1 cycle. So it was a negative study, but a very important study. And this immunogenicity problem of immunotoxins has limited their development for solid tumors. About 25 years ago, there would be meetings specifically on immunotoxins. A lot of pharmaceutical companies were making immunotoxin, but everyone gave up on them. I think we are the only group, maybe there's some other group, that continues to work on immunotoxin. And the previous efforts to overcome this problem have failed, and it has been felt to be a huge problem that cannot be overcome. However, our group has focused on addressing this problem by taking a couple of different approaches. One is that to prevent human immune response to immunotoxins, and second, to make an immunotoxin that is inherently less immunogenic. We and others have in the past looked at different ways to prevent the human immune response to immunotoxins. People have used steroids, cyclosporin, single-agent Cytoxan. They don't work. Patients still develop antibodies. And we did a study with rituximab with a different immunotoxin many years ago, and although we got rid of all the B cells, they still develop antibodies. Fortunately, about 5 years ago, we had a departmental seminar, and we didn't have a speaker, so we tried to figure out who we could coaxed into coming for the seminar. So we called our colleague Dan Fowler, who is a medical oncologist but does bone marrow transplant. So he came to our lab in LMB and gave a seminar and showed that a regimen of pentostatin plus Cytoxan to deplete both the T and the B cells results in host immune depletion without myelosuppression, and that it was safe in patients with renal cancer. So that was pretty interesting. So the next day we met with him and wanted to see in our lab whether this would work with our immunotoxin. So working with Masnari Anda and Musaba in his lab, we looked at the effect of these two drugs on the development of antibodies to SS1P. So we took immunocompetent mice, BALB/c mice. We immunized them with SS1P weekly for 9 weeks. So this is a very immunogenic schedule. And all the mice developed antibodies to SS1P. However, if you pre-treated the mice with pentostatin and Cytoxan, they did not develop antibodies. So this was a very amazing result. We had never seen anything like this. So that was exciting. So within 8 months, we initiated a clinical trial to look at the effect of SS1P with pentostatin and Cytoxan in patients. And the rationale was that depleting the host T and B immune cells with pentostatin and Cytoxan could decrease the antibodies to SS1P. We know that pentostatin by itself has no efficacy in solid tumors, and we did studies showing that mesothelioma cells are not sensitive to pentostatin, and in a prior clinical trial, Cytoxan has no anti-tumor activity in mesothelioma. So the patients who were enrolled were previously treated patients with treatment refractory and progressive disease. And our goals were to determine the safety and whether this was feasible to do. And the primary endpoint was actually very simple. We were trying to see if this regimen can reduce anti-SS1P antibody formation. And the protocol schema was very similar to the mouse experiment, where the patients received Cytoxan and pentostatin, and then after a few days they got the SS1P. If after one cycle they did not develop antibodies, they could get more treatment, and again we pre-treated with pentostatin and Cytoxan. In this pilot study, we enrolled 11 patients, 10 of whom were evaluable for response. Majority of the patients were pleural mesotheliomas. And these were heavily pretreated patients with a median of 3 prior therapies when they enrolled on the study. So the adverse events was grade 4 lymphopenia. Despite all that you heard from other immunologists, our job here was to get rid of the T cell, and we wanted to get the T cells below 100 per microliter. So all patients achieved grade 4 lymphopenia. That's what we wanted to do. And some patients had anemia, transaminitis fever. The SS1P toxicities were similar to what we expected, with pleuritic pain, non-cardiac chest pain, and grade 1 and grade 2 symptoms due to vascular leak from the SS1P. As you can see, in all the patients, there was a marked reduction in absolute lymphocyte count without an effect on the neutrophils, which is important because we didn't want our patients to get bacterial infection. And as you can see, our treatment decreased both the CD4 cells as well as the B cells. And depending upon the duration of the treatment, the B and the T cells were suppressed for a long time, but eventually in all the patients, they came back. So we quickly met the primary endpoint of the trial. So as you can see, as I showed you in the phase 1 trial, 90% of the patients had antibodies after 1 cycle. Whereas in this small study, only 2 patients developed antibodies after 1 cycle. So we never seen results like these with other agents to suppress the immune system. But what was even more amazing was the type of responses we saw in this heavily pretreated patients. So this is an updated data. So out of the 10 patients, 3 patients have very significant partial response, as I will show you in the next few slides. And all the patients who had slide 2 of these patients were all getting ready to go to hospice and were not even sure whether they want to go on the clinical trial. All the patients who got the partial response are still alive and doing very well. And what we also saw that a couple of patients, shown here and here, who had stable disease, but when they were re-challenged with chemotherapy, to which they had previously not responded, they had a dramatic response. Just to give you some examples, this was a patient with extensive pleural mesothelioma, had 3 prior chemotherapies. She was in fact progressing while she was getting the Pentostatin and Cytoxan, and as you can see, she continues to maintain a great response. Even at 30 months, she had no change in the CT scan, and but she had some increase in PET, so we just about 4 weeks ago treated her with chemotherapy, but she is doing phenomenally well. This was another patient with widely metastatic mesothelioma, 4 prior therapies. At the time we saw him, he had significant disease. As you can see, mets to the neck, had superior vena cava syndrome, but he got a very quick response after 2 cycles. And this is CT scans of its chest. He has one of the highest mesothelioma tumor burden I've seen. He had disease in his neck, in his chest, in his abdomen, in his pelvis. Very nice response, and very good response at 8 months. And if you look at his PET scan, this was his PET scan before treatment, showing diffuse uptake. In fact, while he was getting a radiologic response, his PET scan was actually worse, but at 8 months he had near complete PET response. Just one more example of a patient with extensive peritoneal mesothelioma who had a delayed response, is showing initial worsening of the PET scan, followed by a response without any intervening treatment in between, so no treatments here. So we were excited about these results. I think SS1P plus Pentostatin Cytoxan is safe. We saw decreases in anti-SS1P antibody formation. We saw dramatic and durable tumor responses in patients with treatment-refractory mesothelioma, and we're now doing a small biomarker-driven study in mesothelioma with pre- and post-biopsies to further evaluate the immune changes within the tumor microenvironment. So after years of working with immunotoxins, where everyone said that these toxins would never work, it's indeed gratifying to see patients who finally get a response. This was a patient I saw in clinic last week who heavily pre-treated 3 prior therapies, getting a very nice response by PET scan. He had huge involvement of the scrotum, penis, whole of his abdominal wall. So this is good, so we're happy with it. However, it would be better to have an immunotoxin that is inherently less immunogenic. So that has been the focus of our lab, especially Ira's lab over the last 4 or 5 years, is to make a better molecule which is not immunogenic. So just to remind you again what SS1P is, it is an anti-mesothelin FV. This is the light chain, the heavy chain. So this is the FV which recognizes mesothelin attached to the P38 Pseudomonas toxin. As I said, it has domain 2 and domain 3. Domain 3 is absolutely important because it inhibits protein synthesis. Domain 2, we have been thinking for the last 25 years that it's important for translocation and going to the cytosol. But about 4 or 5 years ago, a new postdoc came to Ira's lab, John Weldon, who had no idea what Pseudomonas toxin is. So he came with a fresh pair of eyes. So he did an amazing thing, is that he got rid of all of domain 2, which we thought was crazy because we thought the toxin would never work. So he made an immunotoxin where you get rid of almost all of the domain 2, and we called it SS1 lysosomal resistant. But what was very impressive is that you can get rid of the whole of the domain without affecting activity. In fact, the immunotoxin was much better. And removing the domain 2 gets rid of the majority of the main T cell epitopes of the toxin. But more importantly, you could give mice about 8-fold higher dose of the toxin. And in a rat VLS model, there's a model for vascular leak syndrome. It did not cause any VLS. So this was very exciting. So next, so that was good, but there were still B cell epitopes which were in the domain 3. So patients could develop antibodies to this domain 3 of the toxin. And this is a lot of work done by very talented people in LMB. So they spent a lot of time to identify what are the B cell epitopes which are present in domain 3, and there were 7 major epitopes. and these are hydrophobic amino acids, so they were mutated to alanine, and this is what we call SS1-LO10. By doing this, we remove all the human B cell epitopes and 2 of the epitopes in the domain 3, and it retains its cytotoxic activity but is less immunogenic in mice. So this is very good, but the problem here is that this molecule is about 44 kilodalton because we have got rid of domain 2, and we were worried that this molecule could get easily filtered through the kidneys. So working with Roche Genentech, we made a new molecule where we replaced the Fe with an FAB. This is a humanized FAB to mesothelin linked to this modified toxin. So all known human B cell epitopes and major T cell epitopes of PE have been removed or mutated. The antigenicity is reduced about 1,000-fold compared to SS1P. So if you take patient's serum which has antibodies to SS1P, it reacts about 1,000-fold less. And we can give much higher doses than SS1P to mice, and it does not cause vascular leak syndrome. Data that I won't show is that we have shown that it is has cytotoxic activity against several primary and established cell lines that highly express mesothelin, has single-agent activity against mesothelin-positive tumor xenografts, and marked synergy with chemotherapy. And we have established a CRADA with Roche Genentech, and hopefully we'll start treating patients end of this year. Just to show you briefly a couple of minutes of data with this molecule using primary mesothelioma cells. So these are cells that we established from our patients, NCI M19 and 21. Both these cells highly express mesothelin, about 40 to 50,000 mesothelin sites per cell. And this is the IC50. Shown in the blue is SS1P, and in the green is the Rg compound. As you can see, the Rg compound is significantly more active than SS1P against these primary cultures. We also see marked synergy between Rg778 and chemotherapy against primary mesothelioma cell lines. So we have seen, surprisingly, that mesothelioma cell lines are very sensitive to paclitaxel, which it goes against the published data. There was a phase 2 clinical trial with paclitaxel which showed a 0 response rate. But we see that these cell lines are very sensitive, and also to nab-paclitaxel. And this is a combination study in vitro where we treat cells with either Taxol, nab-paclitaxel, or the Rg. And when we use the two in combination, you see marked reduction in cell growth. And we get the next one. And this is again photomicrographs of cell cultures, untreated, treated with the Taxol, Rg, and if you treat the two, you get almost complete cell death. We have also shown that there is marked in vivo synergy between Rg and nab-paclitaxel against mesothelioma tumor xenografts. So this is again what we call as NCI16 cell from a patient with mesothelioma. And these mice were injected with the NCI-M16 cell line. When the tumors were about 100 millimeter cube, they were randomized to either no treatment or treatment with the Rg compound alone for a couple of doses, or to nab-paclitaxel. However, if you treat the two in combination, you get complete responses, and 5 out of the 7 mice are still in complete response. So this is really very good synergy between the two. So to summarize, anti-mesothelin immunotoxins, SS1P with pentostatin and Cytoxan has produced significant tumor regressions in treatment-refractory mesothelioma patients. Our new generation Rg778 immunotoxin could potentially have more efficacy because it can be given at higher doses for more treatment cycles and hopefully have fewer side effects. But there are other approaches to target mesothelin besides the immunotoxin, and we have been doing some of those trials. One of them is an antibody to mesothelin. This is a chimeric antibody to mesothelin developed by Morphotek, also called MORAB009, which kills cells by ADCC as well as by inhibiting the interaction between mesothelin and CA125. In a non-randomized phase 2 study, this antibody showed increased in overall survival, and there are plans to test it out in a larger randomized trial. I talked about the immunotoxin, antibody drug conjugate. There is a clinical trial going on by Bayer that we are participating in patients with mesothelioma expressing cancer. There's also a Genentech antibody drug conjugate for patients mostly with ovarian cancer. You heard a lot about the chimeric antigen T receptor from Steve and Edmund Moon. There are a trial going on at NCI by Steve Rosenberg and the group at Penn. And the other approach, which is different from these antibody approaches, is to use mesothelin as a vaccine. And there is a vaccine currently in clinical trial. I talked about it yesterday, called CRS-207, which is Listeria monocyte genes expressing human mesothelin. And some of this work, there was pioneering work done by Elizabeth Jaffe at Johns Hopkins, who showed that in patients who received a pancreatic vaccine called GVAX, the patients who were long-term survivors had an anti-mesothelin immune response. And so a lot of work done by Adura, Dirk Borchardt showed that Listeria could be a good immunogen. And I presented the data of the ongoing combination study last week. We believe that our therapies targeted to mesothelin could be useful to treating many common solid tumors because they highly express mesothelin. And there is already hints of that. In pancreatic cancer, this is a clinical trial, randomized clinical trial of the mesothelin vaccine CRS-207 in patients with refractory pancreatic cancer who were randomized to receive either the GVAX alone or GVAX with CRS-207, where there is clearly a separation of curves. So this is very exciting in pancreatic cancer, and there's now a larger trial which is ongoing. So we believe that our work over the last many years has validated mesothelin as a target for cancer therapy. We have done proof of concept studies in mesothelioma, and I think showed for the first time that immunotoxins can work in solid tumors. We also now plan to focus on common tumors, including lung and pancreatic cancer, besides mesothelioma. And we believe that key collaboration with industry have helped us to develop these mesothelin-targeted therapies for the patients. So that has been really very helpful to take it forward. And Ira and I recently described our work over the last 20 years on the discovery of mesothelin and exploiting as a target for immunotherapy. So this work would not be possible because a lot of people working on it over the last many years, including people in my lab, in Ira's lab, collaborators at NCI, and over various different collaborations. And finally, we're always very thankful to our patients who decide to participate in clinical trials, knowing fully well that these therapy may or may not work and all the inconvenience of being in a clinical trial. But I don't think we would make any progress without that. And finally, I've been very fortunate to work with a very nice group of folks, my clinic team, my two fabulous nurses, Yvonne Mallory and Lisa Bankston, and our nurse practitioner. I really don't know what they're doing, but they gave me a story what it is, and folks in my lab who did most of the study. Thank you very much.